Well, good evening, everyone. I'd like to welcome you to the University of Melbourne's Innovations for Regenerative Medicine Translation webinar event tonight. We are about ready to start, so thank you very much for joining us. My name is Andrea O'Connor, and I have the pleasure of being the MC for our webinar panel discussion today. Before we proceed, let me introduce a little bit about myself. So I'm the head of the Department of Biomedical Engineering at the University of Melbourne, and I lead the tissue engineering group there. So my research interests focus on the design, synthesis and fabrication of biomaterials, porous materials and antimicrobial nanomaterials. I have particular interests in strategies for scale-up of tissue engineering, including vascularization and design of antimicrobial materials for medical implants. Before we begin the webinar, I'd like to acknowledge the traditional owners of the land on which I am joining this webinar, the Wurundjeri people, who are also the traditional owners of the land where the University of Melbourne is located. And I pay my respects to their elders and families, past, present and emerging. So thank you very much for joining with us tonight. We have some industry leaders and academics joining us from across the globe on the topic of innovations for regenerative medicine translation. So today's discussion is being delivered remotely, as you're aware, and I'd like to take a moment to thank you for joining our webinar. So I'm pleased to be joined by our panel members tonight. Dr. Daniel Heath is a Senior Lecturer and Assistant Dean of International Partnerships in the Department of Biomedical Engineering at the University of Melbourne. Professor Sarah Cartmore joins us from the University of Manchester, where she is a Professor of Bioengineering and the Head of the Department of Materials. Dr. Killian Kelly is the Chief Operating Officer for Cynata Therapeutics. And Professor Megan Munsey from the University of Melbourne is the Deputy Director of the Centre for Stem Cell Systems and a Professor in the Melbourne School of Medicine. So I think you will agree it's a very interesting panel, and I'm excited to start the discussion. Before we continue, I'd also like to introduce you all to Zara, who is our graphic recorder, and she'll be doing a live sketch of the event today. She'll appear on your screen throughout the session so you can watch the illustration come to life. Thanks, Zara. In this webinar, you'll discover more about limitations and opportunities in regenerative medicine and how we're translating research into clinical applications and industry solutions. Specifically, we'll discuss the need for innovations in biomaterials design and manufacturing and the identification of reliable, well-characterised and ethically sourced cells. So this is obviously a very broad topic area, and I'd like to just introduce a little bit of the motivation and scope for the field. So if you think about the human body, it's a very complex system. And there are many aspects that can cause us problems in our lives due to trauma, genetic defects as we age and as a result of disease. And so regenerative medicine and tissue engineering is one strategy we can use to try to replace and restore the function of tissues that may not be working well in the human body. And there's some great examples of success in this. So something like a hip implant or a total knee replacement is a very successful medical device strategy to replace tissue that is not working well in a particular person. But of course, that's not the same as replacing the tissue with your own natural bone. And there are many examples where we actually don't have ideal medical devices to replace tissue function. So this is where regenerative medicine and tissue engineering might play a very significant role. So there are a number of targets that have been investigated for regenerative medicine. Some have even gone through to clinical studies. So the image on the upper right here is from a study we did on breast reconstruction with Wayne Morrison's team at the O'Brien Institute here in Melbourne, growing vascularized soft tissue for breast reconstruction. But regenerative medicine and tissue engineering has a very wide range of potential applications, including microfluidics and lab-on-a-chip type strategies for high throughput and personalised drug screening, even going to potentially engineering replacement food sources through engineering meat, and one day perhaps engineering organs such as kidneys. So there's a lot of scope, and today we'll just touch on some of the aspects of that and give you a flavour of where some of the challenges and progress might be. So the panel will provide a brief presentation each before we open the Q&A with any questions from the audience. So our panel will be answering live audience questions, and I encourage you to engage with the panel through the Q&A box within Zoom. So you'll find the Q&A icon on the panel at the bottom of your page, depending on your screen setup. And you can submit your questions as we go along. Simply open the Q&A chat box and type in your anonymous questions. And we'll try to get to as many of those as we can, in addition to some that were submitted as you registered for this event. I'd like to note that this event is being recorded, so the video of this webinar will be put on the University of Melbourne School of Engineering MedTech webpage. Okay, let's move to our panel. So firstly, I'd like to introduce Dr. Daniel Heath. Daniel is a senior lecturer in the Department of Biomedical Engineering at the University of Melbourne. He's a biomaterials scientist, and he and his group strive to improve medical outcomes by investigating how materials interact with biological environments. So this fundamental knowledge is then used to develop improved materials for use in next generation biomedical devices. So welcome, Daniel.
Hi, good afternoon, everybody. Thank you very much, Andrea, for the kind introduction. And I'd be really grateful to tell you a little bit about the work that we're doing at University of Melbourne in regard to biomaterial science. If we can go to the next slide, Mel. Awesome. So first off, let's give a general description about what we mean by the term biomaterials. And essentially what we're talking about in this context is a material that contacts a biological system for medical applications. It could be something as everyday as a contact lens, or something as advanced as a hip replacement or a tissue scaffold. And like all material systems, biomaterials have to meet certain constraints for them to work. So obviously a contact lens has to be transparent, but it also has to have favorable interactions with the biological environment, such as good transport of oxygen to the eye. Additionally, in other applications, we need to limit inflammation, we need to limit from thrombosis and fibrosis, and prevent infection. And one thing that I think is really exciting about new biomaterial development is that we're actually designing materials that give instructions to the cells and to the biological environment on how to function, how to heal, and how to improve. Next slide, please. So in my lab, we work with a wide variety of different types of biomaterial systems. So on the left here, we have what we refer as fully defined biomaterials. So these are systems that we synthesize from scratch, often using medical grade materials. So that way we know exactly what is in the material and how it functions. However, I'm a strong believer in that the natural tissues that surround our cells, the extracellular matrix, really make ideal biomaterials. So we also work with decellularized tissues and cell-secreted materials. So this is where we either take a biological tissue, in this case the placenta, and decellularize it and use it to create things like hydrogels that have a lot of biological information in them, or where we take cell cultures, decellularize those to produce cell-specific ECM materials. Next slide, please. So while there's a huge number of main challenges in the biomaterials field, a few of the things that we look at in my lab are technologies that enable large-scale stem cell expansion, materials with superior blood contacting properties, materials that leverage biological mechanisms to direct cell behavior, and specifically, we try to focus on technologies that can be produced at industrial scale, can be commercialized, and can receive regulatory approval. Fantastic, thank you.
Thank you, Daniel. That's a great introduction. And next I'd like to introduce Professor Sarah Cartmell, and particularly to thank Sarah for getting up early in the morning in the UK to join this webinar. Sarah is a professor of bioengineering and the head of the Department of Materials at the University of Manchester. She joined that university in 2014 and she received her Bachelor of Engineering in Materials Science with Clinical Engineering and a PhD in Clinical Engineering from the University of Liverpool prior to that. She was a postdoc at Georgia Tech in Atlanta for 2 years, and she also worked at Keele University, where she was a postdoc, then a lecturer and senior lecturer in orthopedic tissue engineering. So Sarah, it's a great pleasure to have you with us, and I'll hand over to you.
Many thanks for the introduction, Andrea, and thank you for the invitation to be here on the panel. It's a great pleasure. Next slide, please. So my own research interests are in the area of biomaterials and tissue engineering. And more specifically, as you can see in this top blue box here, I have a focus on orthopedics, which includes bone and cartilage, tendon and ligament tissue engineering. I also have a portfolio of work on wound care products with a focus on treatments for chronic wounds, such as diabetic ulcers. And recently I've been working with the Manchester Cancer Research Centre, transferring some of this technology to develop a lung cancer model that can be used for early biomarkers for early lung cancer detection. To do this, my lab has expertise in developing and testing different biomaterials, which include electrospun polymers, as can be seen on the top here, hydrogel systems, composites, etc. We're also interested in different types of stimulus applied to cells in order to influence their activity, be that proliferation, differentiation, or excellular matrix production. And this stimulus includes electrical stimulation and application of specific mechanical regimes. In order to apply these stimuli and grow 3D tissues, one needs to control the environment. And in order to do this, I have a large body of research designing different types of bioreactors. And here you can see a schematic of a capacitive electrical bioreactor system, excuse me, and a co-culture bioreactor. Below, the image that you can see below is to grow an osteochondral piece of tissue. And lastly, my lab is interested in developing new methodologies to provide information about the tissues we're trying to create. So I have a research portfolio developing computed tomography imaging to either track live cells through scaffolds or provide 3D high-resolution structural information on soft tissues. And the example that you can see here just at the bottom is a CT image of a porcine anterior cruciate ligament that we've imaged and been able to track different individual fascicles through the structure. Next slide, please. I wanted to show you one example of a medical product that we're currently translating through my lab. This is a tendon repair product, and you can see here our focus is on hand tendon injury, where we have over 30,000 hand tendons treated annually in the UK. And these are usually trauma cases that present at accident and emergency, and the surgeon will suture these tendons together using different types of suture knots, as can be seen here in the blue cartoons. The knots such as the Kessler knot, the Bonnell or Savage suture knots. And the first 3 months of repair is really important, and currently a quarter of these repairs have poor outcome, with 7% requiring surgery again. We've developed a product that augments this current suture repair that offers a bridge to aid healing tendon. Next slide, please. So the product, which we've recently patented, is an electrospun polycaprolactone device. You can see just at the top here with this SEM image, we have very small fibres that the cells recognise as similar in structure to naturally occurring excellular matrix morphology. And when we twist these fibres, and we can create a yarn, as you can see there at the top middle, which we then knit together to create the device on the top right. The image to the bottom left just demonstrates how we can use a hollow needle to easily deliver the product to the sutured tendon. On our data that we've gained has shown an increase in strength at 3 weeks with this product in place, around about 3 times increase in strength, with it also an increase of excellular matrix proteins and reduction of inflammation. And this product degrades with about 50% remaining after 12 months. And we're at the stage of moving to clinical trial with this product. Next slide, please. And just one more click, please. Thank you. So the last slide I wanted to show you was about a new UK government investment of over £200 million into the Sir Henry Royce Institute for Advanced Materials. This UK institute involves many partners, as you can see listed on the slide, and is led by the University of Manchester. You can see listed here the different themes the institute focuses on, and I'm the UK champion working with colleagues such as Dr. Johnny Blaker at the University of Manchester to lead the biomedical theme you can see here as well. We have had investment not only for the building that you can see and the image on the left, which is where my lab is based, but also over £10 million of state-of-the-art equipment, which is specific for biomedical materials manufacture, testing and characterisation. And we're currently working with our partners and stakeholders to ensure that our landscaping in the field is relevant and fit for purpose for the UK and also internationally competitive. With that, I will close and hand back over to Andrea. Many thanks.
Thank you very much, Sarah. That's a very exciting story about your product translation, and the Royce Institute is also very exciting. So I look forward to hearing a little more about that in our Q&A. But now I'd like to move to introducing Dr. Killian Kelly. Killian is the Chief Operating Officer at Cynata Therapeutics, which is an Australian clinical stage biotechnology company focused on the development of novel induced pluripotent stem cells, or iPSC, based therapies. He's previously held positions in Biota Pharmaceuticals, Mesoblast, Kendall International, Amgen and AstraZeneca. His education was a degree in Masters in Pharmacy from Robert Gordon University, Aberdeen, and a PhD in Pharmaceutical Sciences from Strathclyde University in Glasgow. He's a member of the Royal Pharmaceutical Society and the Australian Institute of Company Directors, and also the International Society for Cell and Gene Therapy. So Killian, welcome to the presentation.
Thank you very much, Andrea, and thanks for the opportunity to join the panel, and thanks to everybody for joining us this evening as well. So Cynata is focused on the development of mesenchymal stem cell or MSC-based therapies, with a bit of a difference, which I'll tell you about in a moment. But many of you will probably be familiar with MSCs because they've been investigated fairly widely over the past 10 to 15 years in clinical trials for a wide range of conditions. And as you can see from the graphic here, the number of new trials just continues to grow year on year. The reason for that is really that there's a fairly substantial body of evidence now showing that MSCs have therapeutic potential in a wide range of conditions because they can modulate the immune system, regenerate damaged tissue, and accelerate recovery from the effects of disease or injury. However, there are major challenges with manufacturing these cells consistently and at commercial scale using conventional approaches. So we can go to the next slide, please. So at the top here is a bit of a representation of a conventional MSC process. So it involves isolating cells from donors. Donated tissue might be bone marrow or fat. Then they isolate the cells, expand them in culture in vitro, and then administer them to patients. But some of the challenges are that there's a lot of variability between donors. And also when you extensively expand these cells, they change. So they tend to enter senescence and lose potency and so on. And we're aiming to address that by using iPSC technology. So iPSCs have the ability to reproduce themselves to an effectively limitless extent, which means we don't need new donors, so we avoid interdonor variability. And that also means we don't need to expand at the MSC stage a great deal, so we avoid some of those problems too. So next slide, please. So we have completed a clinical trial in a condition called graft versus host disease, which went very well, and it was recently published in Nature Medicine, published online in September. I think it will be in this month's issue in print. And that was obviously a great achievement, and it was really a recognition of the fact that this was the first completed clinical trial worldwide using iPSC-derived cells in any disease. And as I said, the results were very good, both from an efficacy and a safety point of view. I don't really have time to go through them now, but you can obviously read our paper when it comes out. And we can go to the next slide, please. So we've also been developing our product for a broad range of other conditions. So we've conducted preclinical studies in a range of different animal models. And in addition to GVHD, we now have clinical trials approved and up and running in a couple of cases in patients with COVID-19 and respiratory distress, critical limb ischemia, and osteoarthritis. So there's a lot going on with this technology, and we will be moving forward with a lot of these programs in the years to come. Thanks.
Thanks, Killian. Very exciting to see those results come out and to see what you're doing. Next up, I'd like to introduce Professor Megan Munsey. So Megan is an internationally recognised biologist, and over the last 25 years, she's made a significant contribution to policy development and community engagement in stem cell science and regenerative medicine. She's the Deputy Director of the University of Melbourne's Centre for Stem Cell Systems, a professor in the School of Medicine here, and she holds senior roles in several national and international professional bodies. Megan received her PhD and Masters of Reproductive Sciences from Monash University and her undergraduate degree from Queensland University of Technology. So welcome, Megan.
Thanks, Andrea, and hi, everybody. First slide, thanks, Mel. So we've heard some fabulously exciting work discussed tonight, and I bet you've seen headlines like these. These are a collection of some of my favourites over the years. And why I wanted to raise this is that I think it's really important that we talk about this gap between perhaps what the community think we can do and what we're doing. And unfortunately, too often it's left to the headlines, it's left to the newspapers to perhaps break the stories to the general public. And there's often a distortion around what we're doing, why we're doing it, and where we're at in terms of delivering on what is a very promising area of science. You'll also see from this collection, it's not just growing perhaps a replacement organ. It's the idea that these cells, and here I'm thinking particularly around stem cells and cells derived from them, that can somehow be used as a replacement cell, that have almost a miraculous property where the cells can, do whatever is required in that patient. And you can see some of the headlines here are carrying the stories around these great hopes that the patients hold. You might see that Michael Schumacher apparently had stem cell treatments that was promoted last year that was supposed to help him with his brain injury. Next slide, thanks, Mel. If you're intrigued as a patient or perhaps as a loved one of a patient about the possibility of stem cells, it's been far too easy to hop online, type in stem cells for whatever condition you're interested in, and come up with ads like these. So while Gillian was just talking about the work that they hope to do around a phase 3 study using their well-defined product in clinical trials for osteoarthritis, you can already get treatment here, and you have been able to do that for some time. You can also get treatment abroad for a whole range of different conditions, including osteoarthritis, but many other applications. And unlike the careful research that my colleagues have alluded to, this work is often decoupled from research, and it's very, very unclear what cellular products are being put back into the patients, and it's often done without any regulatory oversight whatsoever. Next slide, thanks, Mel. So what I've been really interested in my work is this kind of integrated approach to how you strike the balance between enabling innovation and also, I suppose, continuing to give hope to patients. Next slide, thanks. Or next click, thanks, Mel. So I do work around these 4 kind of key areas. I've been working closely with international bodies like the International Society for Stem Cell Research, as well as the International Society for Cell and Tissue Therapy, to develop a series of guidelines that kind of set the boundaries around what we should expect in terms of ethical and responsible translation of promising science. When it comes to regulatory oversight, I've also been working, thanks Mel, with the TGA and other regulators to make sure that the regulations we have in place are fit for purpose. That ad I showed you a moment ago that promised I think a 99.9% success rate for osteoarthritis was a product that hadn't been approved by the regulators, yet was offered and sold in Australia. Why was that the case? And I haven't got time to go into it now, but basically there was a gap in our regulatory framework that allowed these practitioners to work without appropriate oversight. Subsequently, that has now been addressed, and I'm very happy to talk about that a little bit perhaps later. The other part of my work that I wanted to mention is that I think it's really important that we understand what people think and why they think it. And so I've done a lot of very interesting interdisciplinary work with colleagues from sociology and bioethics, trying to look more around collecting from empirical data a perspective of what different people within different publics within the community think about this field. Final click, thanks Mel. And I think it's really important that we take those learnings and integrate it into whatever we do in terms of public education. Obviously, I advocate that we as professionals need to get out there in the community and talk about our work. Don't leave it to the journalists to interpret our work. We need to be out there. But I think we also have to make sure that we're not assuming what the community know. We need to have some evidence to support whatever we're doing in terms of engagement. So I'll leave it there, Andrea, and hand back to you. Thank you.
Thank you very much, Megan. All very good points and really important for us to be thinking about in this context. So now you've heard a little bit from each of our panellists. I'd like to welcome them all back to the screen to join us. And before I turn to the panel, I'd like to encourage the audience to please take the opportunity to open that Q&A box. You can start submitting your questions to the panel. And if you have already given us some questions in the registration process, we'll collect those, and we're also going to try and address those as well. So perhaps I can start with Daniel. Daniel, you mentioned that you're working on extracellular matrix-based biomaterials. So can you tell us a little bit about why creating ECM mimics is such a good strategy for next-gen biomaterials?
Definitely, Andrea. That's one of my favorite topics, actually. So it turns out that the extracellular matrix has been co-evolving with cells and organisms for millions of years, ever since there's been like multicellular life for the most part. And as such, it contains a huge amount of biological information that regulates how the cells behave. And while that ECM is vastly complicated, what a lot of us try to do as biomaterial scientists is to understand how these ECM materials promote specific cell behaviors and then make synthetic materials that mimic those. Because the ECM, while it is amazing, is vastly complicated, very hard to produce, and kind of hard to get a hold of because most people want their tissue. But if we could figure out how it works, incorporate that into a synthetic material, and then commercialize that, we could make some really amazing biomaterials that really push the body to heal or to function naturally.
Thank you. Yes, biology is so complex, but we do our best to try and mimic some aspects. Sarah, can I move to you, and maybe could you elaborate on what you consider sort of the main areas for research investment, particularly in the UK context? You mentioned the Royce Institute. What are the sort of focus areas for investment in the UK in this field?
Absolutely. Yeah, so through the Royce Institute, I've been able to work with over 200 different academic and industrial stakeholders. Can you still hear me okay, Andrea? I think my video is frozen. So I've been able to work with over 200 different academic and industrial stakeholders in the biomedical materials industry. And as it is, and I'm sure in Australia, it's a rapidly growing field which needs to address our growing and ageing population. Our landscaping activities that were performed through the Royce has showed a need for investment into fibre technology, bioelectronics, additive manufacture, which includes increasing rate of production, resolution that we can print, what we can print, for example, different composites together. We need to focus more on minimally invasive surgical products such as injectables, and also a focus on scale up. This particularly came from industry stakeholders. So this isn't only just increasing the rate of manufacturing testing, but also better methods of manufacture, which would increase reproducibility and reduce wastage. But yeah, they're the main areas that we're focusing particularly through the Royce Institute.
Fantastic. And does the Royce Institute funding have a particular timeline to try and achieve those goals?
We do have, a 5-year, 10-year plan. We've just submitted our sort of a years 6 to 10 bid through to the government again, which is currently being considered. We've got particular big ideas that we want to focus on, which we'll be looking at. Obviously, I know there's a topic on COVID right now in terms of antimicrobial, so it's not just viral, but antibacterials and things like that as well. And so all of this fibre tech, bioelectronics, additive manufacturing that I've mentioned could all fit into those big ideas. So we're looking more at challenges rather than, obviously we've got the infrastructure in place now to improve those particular areas, but now we're looking at particular needs and challenges.
Fantastic. That's hugely exciting. Killian, can I maybe come to you? And it's interesting that Cynata is an Australian company commercialising technology. The technology, I believe, came from the US, and your first clinical trial was in the UK. Can you talk to us a little bit about why you've got that sort of landscape?
Yeah, absolutely. So the technology, as you said, arose from the US, from the University of Wisconsin-Madison. The story how Cynata came to be in Melbourne really was, I guess, a matter of the scientists in Wisconsin having a connection with an entrepreneur in Melbourne, and they decided to establish the company here. And so it's probably a bit unusual. We tend to see Australian technology going overseas rather than the other way around a lot. And why we ended up doing a clinical trial in the UK, it was largely down to the regulatory pathway. So our technology was very novel. Nobody had done a clinical trial with iPSC-derived cells before. So we really had to go out and talk to regulators worldwide to try and figure out what their expectations were and what we needed to do to get a clinical trial approved. We found that the European Medicines Agency, or the EMA, and also the UK MHRA, which is the national regulatory authority there, both had a very, I think, pragmatic and flexible view of this and a real enthusiasm for the type of technology we're developing. So basically, we had a pretty clear and relatively speedy path to a clinical trial there, more so than in other countries. An added bonus was by getting approval in the UK, it also meant we could then get an Australian clinical trial approved under what's called the CTN scheme, as opposed to the CTX scheme, which many people might know is much more onerous and time consuming. So there was an advantage there as well. And that's really it in a nutshell. Yeah.
Right. That's very interesting. Thank you. Megan, one for you. Regenerative medicine has this great promise to transform future healthcare. But as you touched on, engaging with the community and managing community expectations is really important. How can we do that better as scientists and practitioners in this field?
I think just get out there. I think I just saw in Zara's cartoon that's, I think, a phrase that she didn't indicate it, but I do think it's up to us. I think we have a responsibility to think about who our key stakeholders are and work with them strategically. So I've had a lot of terrific engagement with different patient advocacy groups, people who are trying to support those in the community with perhaps multiple sclerosis or with Parkinson's disease or with osteoarthritis. And whilst my message is quite similar to each of those groups, it's nuanced depending on that condition. And so I think the onus comes back onto the researchers and to industry to do that well. I think the other area that I think we could do a little bit better is working with media. There's no denying, even though we're in, Web 2.0, there's no denying that media coverage still does have the capacity to transform people's views. So I think we need to be careful about how we talk about our work to research, to the media, to journalists. And when there is something that perhaps has been misrepresented, let's have a conversation about it. Let's just not sort of point fingers and say they could get that better. I think, again, let's have a conversation and raise the level of understanding.
That's great advice. As a scientist, if you see something that you think is wrong in the media, how do you engage in that conversation? How do you start that process? Is it just Twitter, or should we be calling the journalists? What do you recommend?
I think it depends on, how I suppose how many steps removed you are. But I think if your work is being covered by a journalist and you've been interviewed and you have a relationship, wow, that's the first place to go. There's also the Australian Science Media Centre, which has this fabulous remit of working with journalists to get science right in journalism. So there's opportunities to perhaps promote yourself as an expert within your space. So that's the starting point. But I also like this, and I think particularly for our students who are coming through and our ECRs, there are so many great community groups to go and speak to. It hones your skills about trying to be able to explain your work and why you do it, which no doubt will help you with grants and all sorts of other things in your life. But I think it's a really important community service. So again, I like a bit of a kind of a buffet approach where you tailor what works for you and your research. But I think, again, we have a series of tools that we should be using.
Fantastic. That's great advice. Now I'd like to maybe come to some of the translational challenges, particularly focusing initially on biomaterials. So I might ask Daniel and Sarah each to comment about what do you think are the key hurdles in translating some of the biomaterials development. So maybe Daniel, if I come to you first of all, I know you're doing work, for example, in stent design, but you might like to draw on that example. How do we get that translated? What are our key challenges?
Definitely. I think by and large, the biomaterials community does a really good job with coming up with new and better materials. I think what the challenge then is to translate that from a lab-based discovery into something that has some form of real-world impact. And that is, I think, where we need to do a little bit of work and development. And for instance, are the materials that you're working with medical grade? Or are you working with medical grade precursors and reagents? Are those even available? The translational process is vastly complicated, very time consuming, and very expensive. So dealing with clinical trials, then moving through regulatory approval and the issue of reimbursement and insurance and whatnot. It can take such a long period of time to make it through that translational process, and it usually takes millions of dollars. So there's a huge financial and time burden associated with that. And then there's some more industry related or engineering related feats, such as can these things be produced at industrial scale in a inexpensive manner? Because at the end of the day, you're going to have a company that is willing to manufacture and make these materials or these devices, and they have to be able to make a profit from it. So if you don't plan ahead of time how you are going to translate that innovation, it's likely not to make it to any real world impact. So that's something we're doing really well at Melbourne, I think, is thinking about what are those barriers to this commercialization, to this scale up, and how can we design our things at the outset? How can we design our research at the outset in order to navigate through those challenges?
Fantastic. And Sarah, I'd love to hear your perspective on that and perhaps your experience with your tendon-based product.
Yeah, absolutely. Totally agree with what Daniel's been saying. I agree. It's not the innovation. I think we've got plenty of this. So I don't think it's that we should stop innovating. I'm not suggesting that at all, but I think we do that very well. I think I agree with Daniel in that one of the hurdles is the translation of that then. And I think one of the hurdles we've got is the researchers we have in the lab perhaps not understanding the roadmap and understanding or considering the correct regulatory input early on. And I think if we, and what we're trying to do with our Centre for Doctoral Training here at Manchester, I know you're doing the same in Melbourne, is to really engage those students and the researchers early on so they understand, the health economic impact, they understand the regulatory, what classification their device is, who they need to connect with, whether or not that's patient input, clinical input, larger industrial connections, the SMEs, and making sure they're having that conversation early so that we're in a position then that it should be smooth and that transition all the way through. And I also agree with what Daniel was saying in terms of the practical aspects of increasing the rate of manufacturing testing, not necessarily at the industrial scale. We need to consider it, but actually the industrial scale isn't too bad. It's actually the pitfall is where we're doing the research stage, and particularly the SMEs that don't have it, which is then translating those products. They don't have access to that size of scale up of manufacture to be able to rapidly be able to get all that testing done. And this is something that the Royce is addressing in terms of that research stage sort of scale up equipment for manufacturing and testing that people can, as a facility, people can come and access without having that initial investment and outlay in the company. I also think we need to invest further in supporting entrepreneurship and startups, not just through financial investment, but it's more through mentoring schemes. I really think that would make a difference. I do apologise about my video. I'm going to try and sort that out.
No problem. That's fantastic to hear. I think it's something we're very interested in Australia, how you do that transition in the scale of manufacture for all sorts of different processes is a challenge. So I'll be fascinated to hear how that goes at the Royce. We have a related question come through from the audience. Catriona's asked about how much of a role patenting plays in this process. So, Sarah, have you patented your technology? Is that an important aspect?
I think it's really important, but my video stops again, but I won't switch it off, so at least you've got a picture. The problem is, as an academic, our metrics are publications, right? And so really you have a contrast. If you're doing work, particularly in an applied area, you want to be able to see it as very successful. A patent is successful, it just takes longer, right? And so if you need to be able to make sure that the patenting is quick, so you need to make sure you have enough data to be able, within that first year, when you first file your patent, to be able to get that out, and you need to know your market. So you need to have that team around you. Obviously, most universities have a very good tech transfer office that is there to offer help. But as a new academic, that becomes more difficult because they don't have that experience. And so I think we really need more help and support for more early career researchers, which is what we're trying to do at Manchester. But I think as a whole, we could do more as a community with supporting the ECRs coming through for that process. money makes the world go round. So obviously patenting is really important. So if you want to translate your product, you really need to have a patent to make that interesting for industry to pick up, which will then help translate through in the UK and the NHS and obviously other healthcare systems. So I do think it's something we need to be considering early on.
Yeah. Andrea, I might
Thanks.
jump in there really quick. One thing that I think is really interesting is that in academics, you have this contrast and this tension between what is scientifically interesting and what is commercially plausible and feasible. So in Sarah's work, she worked with PCL, which is an amazing material, has a really long track record of success. But from a basic fundamental science point of view, it's not the most exciting material. So sometimes we have to kind of dance around what will get us in the high impact factor materials journals versus what can actually make real world impact. So the patenting is important, but also the navigation of that complex kind of academic landscape.
Yeah, Megan.
I just wanted to jump in on that as well and just ask all three of my fellow panelists the importance of clinical engagement. So that was just a great example of that kind of tension between academia and industry. But what about sort of relevancy and feasibility in terms of the clinical context, and how do you manage that and build your relationships to support your research?
Yeah, maybe Sarah, do you want to tackle that one first?
I think it's really important, Megan. We have a clinical pull and we have a technology push in all of our research, and we're very careful that it's not one or the other. We have both innovation going on, and we work very closely with clinicians. And I know you guys do at Melbourne as well. From my correspondence with my colleagues about different projects, it's so important. And it's not just the clinicians; it's the patients as well. Something as simple as a wound dressing with graphene oxide, which is black, and the clinician might say this works great. The patients might say, it looks horrible. I don't want that on me, right? And so you do need to have that engagement about what's important to the end user. And we have that dialogue going on all the time, but everybody needs to engage with that early on. It's not something you need to consider at the end as well. You need to have that constant engagement with the clinical teams.
Killian, what's your perspective from the industry side? I'm sure you're working with clinicians along the way.
Yeah, absolutely, and I agree. It's really important to get the input from clinicians, and I think Daniel mentioned the importance of the kind of what's going to have a real life impact, and often an example that I've had experience with is where we're often looking for unmet medical needs. So you'll be aware of a particular patient population where there are very poor outcomes. So there's an unmet need. We've got a product we think scientifically can help address that unmet need. So that sounds like it's kind of problem solved. But when you talk to the clinicians, sometimes you find out that actually the reason that there's an unmet need is actually not that there are no effective treatments, that there's some other factors at play, and you bringing along a new effective treatment doesn't actually solve what the problem is. And that kind of sounds really counterintuitive, but that actually can be the reality. And so I think until you actually engage with the clinicians who are the ones who will actually not only conduct the clinical trials with these products, but actually ultimately be the people who largely control whether they're used or not when they're on the market, it's absolutely crucial to have that input, and if possible to have it early on, because you don't want to spend too much time and effort pursuing something that's not really going to achieve anything.
Absolutely, yeah. Do you find it easy or difficult to find clinicians really willing to engage in new technologies and try out your new cells? There's a lot of people, I think a lot of clinicians are very excited about new technologies. It's fair to say that that's not the case with all of them. And I think you'll also find, just like any group of people, some of them might be very wedded to what they do at the moment and, very kind of reluctant to change how they practice. And I think especially you get this kind of a challenge where, one of the things we find in regenerative medicine is some of our potential treatments might, for example, be a replacement for surgery. And so if you have to go and persuade surgeons that it's a good idea to pursue something that might mean what they do isn't needed as much in the future, there's obvious kind of, challenges there in getting people engaged. But yeah, I think there's certainly a lot of clinicians who are very excited about new technologies. There's certainly enough of them for us to find them and to engage with them, put it that way. Right. Yeah, that's fantastic. And there's so many tensions in this whole process of getting the right people and the right process. Megan, I wonder if I can follow up with a question for you. Is there a roadmap in Australia for how we might translate regenerative medicine to industry, or if not, how would we go about developing such a thing?
Yeah, so look, thanks, Andrea. It's a great question because I think again Zara's captured this already on her graphic design, but this issue of a roadmap is important, and a lot of people are thinking about it. And I just want to draw your attention to a recent announcement from MTP Connect around a new sort of catalyst body that's going to look at doing exactly this, to draft a roadmap that will perhaps address or at least facilitate, catalyse progress in this field. So there's been various other initiatives led by MTP Connect and others looking at how to kind of establish this pipeline. What do we need? What is a typical product, and what does that pathway to market look like? And I suppose one little cautionary comment I'd make, and I'm curious to know what Gillian and others think, but typical product for me is a bit of an issue. I think we have to be careful that obviously, tonight we've talked about devices, we've talked about cellular therapies, maybe there'll be a combination of the two, and each of those will have perhaps a slightly different pathway. So I think I'm really excited about this new initiative that is looking to develop this roadmap. So I say stay tuned, visit the Oz Biotech and MTP Connect websites to read a bit more, but also learn a bit more about the MRFF stem cell mission that I have to say conflict of interest. I'm an advisor to the Minister and to the Department on this. But, there are these really great Australian government initiatives to fund area in this research. And in the case of the MRFF, it's really looking at discrete projects that are trying to take and meet these unmet needs using regenerative medicine. So lots of great initiatives. And I think, in Australia, we've been very fortunate to have a long-term commitment from various governments around this field. So I don't think it's simple. It hasn't been solved yet, but it's certainly under, a watching brief. And so to keep looking, but I don't know if there'll be one pathway that fits all.
I'm sure there won't be. It's so complex. For our overseas colleagues, the MRFF is a scheme in Australia, the Medical Research Future Fund, where the government has invested large amounts of money, and a lot of that funding is focused on translation. So aiming to translate some of the medical research outcomes to clinical impact. So that's what Megan was referring to. There's a mission there around stem cells particularly. So Sarah, I wonder, in the UK, is there a sort of road mapping exercise about this translation, or is it a bit more focused on institutes like yours?
Yes, indeed there is. And regenerative technology, we have the cell and gene therapy catapult in the UK, and we have some other catapults which are there to help assist the translation, not just of academics, but SMEs as well, through to the clinic. We also have the regenerative medicine platform that's led through Molly Stevens at Imperial College. So that's the academic kind of push from that side. And of course, we have various reports that are guiding all of us through this. We have, the House of Commons report 2017 in regenerative medicine technology. And we have various institutes, such as the Institute of Materials Mining Metallurgy, which has a biomedical section, which obviously commissions different reports as well, which brings together all those sort of expert key opinion leaders just to say this is what's needed right now, which is essentially what we're discussing today as well, all the same sort of topics that are coming up.
Well, it's good to know we're on the same page. Megan, do you think that we have the appropriate regulations in place currently? And we have an audience question related to that about how the general public get their information. So do you think that we're doing a good job, or, the scientific community is sharing information appropriately and influencing regulations appropriately?
Look, I think that there has certainly been a change recently, which I think we should celebrate, and that's seeing the Therapeutic Goods Administration take a little bit more of an active role in the use of the patient's own cells. That was the little regulatory ambiguity I alluded to earlier. It has been possible under sort of effectively a practitioner's exemption for a doctor who's registered here in Australia to be able to use a patient's own cells without any oversight. And again, as I mentioned earlier, it's often decoupled from perhaps expertise around cellular manufacturing and processing and even sort of perhaps understanding the basic biology of those cells. So we have seen a tightening of that regulation. We're yet to see whether it's effective. Whilst that ad I showed you has gone, I could point to several others. These are clinics that are operating in shopping centres who are selling products for about $10,000, whether you have dementia, whether you have osteoarthritis. So I don't know if we've got it quite right yet, but I am I'm a little bit more hopeful that we have the right mechanisms and the right people thinking about it. The question around community understanding, I think it's very hard to ignore those ads. So whilst there is direct-to-consumer marketing, I think we'll have an ongoing battle. I think whilst there are people who have unmet needs, they're going to look. So what I want to keep reinforcing is that we need to have a conversation. People looking and trying things, I don't think we should demonize them or belittle that. It's a natural reaction to having a complex condition. But what I would like to think that what we're doing is trying to highlight where we are on that translation and perhaps manage people's expectations. And again, I think that's through the engagement mechanisms that I mentioned earlier.
That's fantastic. There's a related question come through from Simone in the audience about ethical concerns, and she's asking, does that slow down the translation and commercialization process? So how do you manage that tension, do you think?
Well, again, as I alluded to earlier, I think we have to have very clear guidelines around what is ethical practice. I think there's still a lot of conversation and confusion in the community around, the use of human embryos. For me, having worked in this space for decades, I find that conversation something that I feel like it's something we had many years ago. But, look at the controversy only a couple of months ago when there was a comment made by the Prime Minister about making a certain vaccine, if it ever arose, for COVID-19 mandatory. And that vaccine involved the use of a particular cell line that was derived from fetal tissue. Now, that created a lot of community agitation. And I was quite surprised that the use of fetal tissue, in particularly those historical lines that we use in research all the time, still is very sensitive. So to answer the question, I think we have to be aware and sensitive to the ethical issues, but reassure the community that what we're doing, as you mentioned earlier in your introduction, we're using material that's appropriately obtained. And where it's not, we call it out.
Yes, I think that's a great point. And I think in general, people wouldn't be aware necessarily of where the cells that we use for vaccine development and other things come from. They wouldn't have been exposed to that.
Yeah, and I think many people don't really mind, but perhaps they want to be asked, or they want to be, at least there needs to be some transparency. So again, sensitivity to what these issues are and being very clear about it and what we do about it and why.
Yes, fantastic. I'd like to switch gear a little bit and talk about some of the technology development that's going on. So Daniel, if I can come to you, we have some questions from the audience around the limitations and pitfalls of developing personalised devices. So what are the challenges in developing personalised tissue engineering strategies?
Definitely. So with regard to tissue engineering, a big one is cell source. So one of the reasons that I'm a big fan of what Killian and Synata do is because they found a way to manufacture these MSCs, which have a lot of clinical applications at a very large scale, which is something that is really challenging for a lot of cell types. And Killian, correct me if I'm wrong here, but the MSCs are kind of unique in that respect because they tend to be either immunoevasive or immunosuppressant, whatever path you want to go with. So they have the ability to be done through an iPSC route. So it's actually not necessarily an autologous cell therapy. But many other cells need to be from the patient donor or someone closely matched. So therefore, it becomes a lot harder to culture cells from, say, a different donor up to large numbers and use it for a new patient. So application and obtaining the cell numbers is critical. One thing that we still can't do necessarily is create well-vascularized tissue engineering constructs. So that means that without the appropriate blood supply, if something that's created outside of the body is implanted, before the blood vessels are able to grow into it, a lot of the neotissue actually dies, and therefore that results in failure of the tissue engineering construct. Another really interesting area that I think is fascinating and has some really good engineering answers is how do you go about creating that kind of microscale topology and patterning and cell structure within these much larger tissue engineered constructs. So for instance, with kidney organoids, they use a lot of self-assembly of cells in order to create those, but they can't get a single ureter. So therefore, even at a really small organoid scale, self-assembly is not necessarily sufficient for the cells to organize into a large coherent structure. So creating fabrication technologies that allow us to recapitulate some of the really unique structures that we see in really complex native tissues and organs is going to be critical for progressing the field.
Thanks, Daniel. Now, we're very close to the scheduled end of our webinar, but we've got a number of questions remaining, so we might continue for a few more minutes. Please do stay with us if you can. Sarah, I'd like to ask you a related question to what Daniel was talking about. From our audience, someone has asked about how you envisage the translation of 3D printing or bioprinting of biomaterials, and how might we get that into clinical and biopharmaceutical applications?
Yes, so I think 3D printing for pharmaceutical and translation. For pharmaceutical, I think 3D printing's got advantages for creating bespoke shapes and dosage forms for personalised medicine and targeted therapy. So additive manufacturing could be used to create, different cold control release products that are profiled for a specific patient need, and indeed might even involve sensor technology where a drug is released given a particular stimulus, which might be, change in pH or chemical presence. And we've already seen that, 3D printing in the pharma sector anyway has been used. The FDA approved its first 3D printed pill, an anti-epileptic drug called Spritam, and 3D printing has actually been used over the years to develop different nanoparticles and solutions, living items such as folic acid and antibiotics already. So we're already seeing this being translated. Are you wanting a bit more information about the actual translational pathway then, you think, on 3D printing, or...?
I think our audience member is interested in that. What are the things that we need to do to actually get it to work in translation?
Well, I think it depends on what you're trying to print. So right now we already have some very good technology that is already being used, right? So it's already been translated through. When you're looking at more complex solutions, it's really looking at regulatory hurdles for the particular market. When I'm saying market, it's not just the medical device or whether or not it includes cells, because you can print cells at the same time as printing materials, right? Or the country you're trying to translate through, whether or not it's the FDA, whether or not it's the EU. We've got Brexit going on. Where I am, so Northern Ireland is going to be following the EU. The rest of Great Britain, we're still not too sure. So, we have to align. And so you have to consider those things. I think that's going to be the biggest. We obviously have some technology advances we need to make, as I mentioned, I think earlier on in the webinar, in terms of reproducibility, scale up, the rate we can print certain things and how we can print certain materials. So we mentioned polycaprolactone before. We can print PCL, but at not a very high resolution. And if you're wanting to look at very high resolution products, we need improvement in the area, as an example. But I think the biggest hurdle is really the regulatory. If you want to have 3D printing in the surgery there, I think there's still a lot of work to be done to get that through and get that safe.
Yeah, I agree. I think that quality control and understanding is going to be absolutely critical. Another challenge that we all need to face in translation is around differences between animal and cell lines, human cell lines, animal models and humans. So, Killian, I wonder if you can give us some insight about how you manage that translation and the uncertainty at each step there.
Yeah, sure. So it certainly is a challenge. I think the first thing that we have to do is simply accept that there are differences between models and humans, and there always will be. And that's really why ultimately we will have to do human clinical trials. Having said that, the models, there are a lot of very useful preclinical models out there, and it is important. It's usually absolutely required to do initial research in appropriate models. I think when we do that and we get encouraging results, I think then the question that needs to be asked is, there's going to be gaps in our knowledge at that point. But the question is, can we fill those gaps by doing more preclinical research or not? If we can, then we should do more preclinical research. But often we can't, and we need to just accept that we can't, and that the questions at that point can only be answered by human studies. And, if we have enough confidence to believe that there is a reasonable prospect of benefit in humans down the track, that it's appropriate to move into humans. But we have to do it very carefully. So, for example, moving into a phase 1 clinical trial, the dose selection needs to be very conservative and the escalation needs to be very conservative. And we also need to design the trials in the right way. So, I think one principle is we should really act as if the investigational treatment is not going to benefit the patients that are enrolled in the trial. Now, we hope it will be, but if we assume that it won't, then what that means is that we shouldn't, for example, withhold existing treatments that we think would benefit the patients. So instead, we would add our treatment to existing treatments, for example, or we might seek to only enroll patients who, for one reason or another, do not have any treatment options at the moment. So in other words, we can be sure that aside from a safety issue, the patient isn't going to be worse off by being enrolled in a trial, basically. Now, the safety itself, as I said, can be managed to an extent by ensuring that you're cautious about how you design the trial. There are other things we build in, like what we call sentinel dosing, which basically limits the numbers of people exposed early on until we have enough data to convince ourselves that it's safe to enroll more patients. And we will use independent data and safety monitoring boards to make sure that if there are any concerns, that they're being reviewed independently and that they're not being overlooked by people who are obviously very close to the project and who want it to succeed. So we have independent people to make sure that if there's any concerns that they're acted on. And I think that's really what we have to do, is just kind of, we have to move forward into human research at a certain time, but it's to, I suppose, be confident that it is the right time and that we're doing it in a kind of a cautious and in a careful way, basically.
Yeah, that's fantastic. Thanks, Killian. We have some questions. I know we have some students in the audience and maybe people who'd be interested in moving into this field. So, Sarah, do you have any thoughts about jobs or skills that our students might not be aware of if they want to work in this field? How would you recommend that they get involved?
So, they'll probably already be aware of research and development opportunities. Obviously in academia, but carrying on in industry. And they'll obviously already be aware of, sales. So you do need a technical understanding if you're going to be in sales as well. But I think one area that's opening up more is kind of a business engagement role where a technical base is needed. Now, there's more and more investment from our governments in really trying to have impact, particularly from the universities, of our work. And I think there's been a change of how industry do their own research. I think 20, 30 years ago that there was more money, right? And so they actually had the majority of the facilities on site. They could afford to buy the testing facility, but now they're kind of outsourcing that and working with universities more. And there's more funding available in the UK. It's through Innovate UK, and also there's more kind of applied translational grants where it's preferred to work with an industrial partner, where there's more chance of success of translating something, if you like. You've already got that roadmap and that route through. There's more funding in the area, so I'm seeing more and more roles where you have a very skilled business engagement where you're still talking science, but it's more about project managing the roadmap and the translation and helping partner all of the interdisciplinary team that I've been mentioning, linking clinicians, linking the patients, helping the clinical trial set up, whereas otherwise it's the person running the project, the academic. And this is where we've had a problem before, because there's we don't have experience in that. That's not our main role, right? And that's what we've been trained to do at this point in our career. We're trying to change that, but I think there's more and more people in those roles to help facilitate that, which I think is quite interesting that people might not know about.
Yeah, I think that's a great suggestion. And I think here as well, those roles are growing and very important. You need great communication skills as one of the aspects to talk to people across different disciplines. Daniel, I have a related question from Stephanie in our audience about how you network outside the research lab. So how do you get to meet people in industry and how do you make those networks?
That's a really good and a really complicated question. So I think I will be quite honest. I got my postdoc position because I stalked a lady at conferences for 4 years.
Nice one.
Nice stalking. Stalking probably a little bit too hard of a word. I went out of my way at every single meeting to reintroduce myself and talk to a particular academic who I was interested in working with, and that worked out very, very well. So I think what I'm trying to say is be proactive in developing your network. So if there are individuals that you have the opportunity to meet, that you think you want to meet, and that could be useful later on, don't be shy to go up and talk with them, because at the end of the day, it's up to you to grow your own network and for you to take initiative in order to make that happen. And I think you would find that most people are quite willing and eager even to talk to people, because they're constantly looking for new talent who's coming up. They're constantly looking for people to join their lab, so on and so forth. So I don't think you should be afraid of getting rebuffed, because again, we all need to nurture the upcoming generation. And part of the way we do that is through networking, and that might lead to mentorship or sponsorship, so on and so forth.
Megan, you wanted to add to that?
Yeah, I just wanted to put in a plug for some of these professional bodies. So AusBiotech, for example, has an annual meeting. They're always very keen to take volunteers to help with, you have to work behind the scenes, but it's a fabulous opportunity to meet people. So going to conferences within your specialty is important, but looking for opportunities to go to meetings and perhaps as a volunteer is a great stepping stone for students.
Great idea. Sarah, you had a comment too?
I agree with all of those suggestions. I also think making use of your mentors, to help introduce you and build your network. And that doesn't just mean you're assigned official mentor. I always make sure, and I still make sure that I'm kind of just like Danny was saying, I'm kind of bugging people, but people are willing to help, and they say, oh, well, I'll introduce you to this person, I'll introduce you to that person. But you do need to tell people. If you're sitting by yourself and thinking, I would like to do that, we're not, going to be able to read your mind. You have to go out and speak to people, and then that's how that networking will help.
I might also jump back in. So if you're a student at Melbourne, the university has a couple of programs that try and help people build networks, particularly with industry. I did one a couple semesters ago called the TRAM Project, which is Translating Research at Melbourne. It's quite full on, so it's quite a commitment, but some master students who were doing their final year projects, some academics and postdocs, would take a technology that they were working on, and the goal is to do 100 interviews with various people who are stakeholders related to that technology. So it might be device manufacturers. For me, it could be clinicians who would be interested in using the technology. So it almost forces you to kind of get out of your shell and really go gung-ho at meeting individuals. So there are programs like that are available. And that helps a little bit, I think, with making connections, because then you have an official program that's branded by the university behind you. So I think it's much easier to get people to engage with you in that manner. So look for opportunities like that are available, because they're becoming much more prevalent in recent years, especially as we're being asked to interact more and more deeply with industry.
That's all great advice. Thank you. I might draw things to a close. Killian, maybe can I ask you one last question? If students or young scientists and engineers are interested in getting into a company like yours or working in industry, do you have any tips for them?
Yeah, look, it's I suppose one of the challenges with small companies like Cynata is that we often operate in a kind of a so-called virtual way. So we're not all in one building and don't necessarily have our own labs. We kind of have work going on all over the place. So sometimes smaller companies like us find it more difficult actually to bring on, for example, interns or even kind of entry-level people. But what I would say is bigger companies often have much greater opportunities there. And even if your interest is, I suppose, and I can say my career, I started out, my first industry job was with AstraZeneca, which, you know, huge company, employs 70,000 people. And I think as I've gone through my career, I've progressively joined smaller and smaller companies as I've gone on. And that wasn't really intentional, but I think that's kind of, in some ways, maybe it wasn't that surprising, because I think the bigger companies actually offer a lot more opportunities for entry-level positions, and they tend to have proper training programs, and there's lots of people you can learn from. And so I suppose I would probably, if I was back at that stage now, I would probably actively seek to do with what I did, which was probably by chance more than anything. But that was to kind of join a big company where there's, you know, really so many opportunities to learn, and then you can move on from there very easily. So you're not by any means tied to the first company you join.
Fantastic. Yeah, I think that's great advice. So I think we could continue talking all night, but we really do need to wrap up. So I'd like to thank you all very much on behalf of the University of Melbourne. Thank you to the panellists for your contributions to this Innovations for Regenerative Medicine Translation webinar. I'd like to thank the audience for your engagement, for coming along, and your questions and involvement in the discussion. Particular thanks to Zara for your fantastic live graphic recording. That's a great memento of tonight's event. And I'd also like to thank Dr. Brooke Ferrugia, who's been here helping with the Q&A in the background. Finally, we would appreciate your input on our post-event survey, which will appear when the session ends. So we do value your feedback. We're planning to run some more events, and we'd love to know what you are interested in. And if you provide your details, we can keep you up to date with the next things that are coming. So thank you all very much, and I wish you good night or good morning if you're in the UK. Thank you very much.